phosphates on it. And so when you put DNA or lyse cells with the DNA in an agarose gel and then put an electric field across it, that electric field, because of the charge in the DNA, will drive the DNA across the electrophoresis gel in a manner that is dependent on the charge on the DNA, which depends on how big the DNA is. So little pieces of DNA come out of the gel into the well, come out of the well into the gel very quickly, and larger pieces are much slower. And so by looking at how far and how fast the DNA comes out of the top of the gel and into the well, you can get a measure of the number of the size of the DNA, which is a measure of the number of breaks in the DNA. And this is just an example of the kinds of data where you can look at how much DNA damage there is as a function of radiation dose. Now, this kind of an assay needs a fairly high radiation dose to get it to work very well in mammalian cells. But another assay that you'll see a lot in the literature, based on the same principle, is single cell gel electrophoresis, the so-called COMET assay. And the idea here is the same thing, but now we look at individual cells. This is the nucleus from one cell that has been embedded in an agarose gel and lysed. And if the cell has been irradiated and that electric field put across the agarose gel will start to make those little pieces of DNA come out. And then if there's more damage, more pieces come out. And with lots of damage, all of the DNA will come out of that nucleus and into the gel. And by quantifying how much DNA is out here in the tail, the damaged DNA out in the bulk of the gel, compared to what was in the nucleus, the head, then you can get a measure of how much DNA damage occurred. And using these kinds and lots of different assays for looking at single strand breaks and double strand breaks, in this review article, we see lots of dose response curves. So we're looking at DNA damage of different assays as a function of radiation dose. And some of these are neutral conditions. So we're looking at double strand breaks. Some are for single strand breaks under alkali conditions. And one of the important things to keep in mind is that if you look at the dose ranges that are used, you usually have to use higher doses to look at double strand breaks than single strand breaks. And that's because there are many more single strand breaks. So there's lots and lots of single strand breaks produced per unit dose compared to double strand breaks. But in most of these cases, these initial numbers of breaks that are produced without repair produce linear dose response curves. But as soon as repair occurs, then you start to get a curvilinear response. And that's shown here in this upper left panel, where now we're looking colony formation. And we'll talk about that more in a later lecture. But this means that these cells survived and were able to continue to proliferate. And so they had to have repaired their DNA damage. And when repair occurs, you get a curvilinear dose response curve. Initial damage is dependent on dose in a linear fashion. Again, something important to keep in mind. One of the assays that you'll see in the literature a lot nowadays is this assay that's called focus formation or foci formation by looking for clumps of DNA repair-related proteins. And the first assay that was developed was for this thing called gamma H2AX. H2 is histone 2A, variant X. And it was discovered that when cells were irradiated, this histone specifically got phosphorylated on a specific protein, on a specific amino acid. And that became known as the gamma H2AX, the phosphorylated histone. And when antibodies were raised to that damage and were fluorescently tagged, it was found that cells labeled with these antibodies would show these clumps these aggregates or blobs of damage, of proteins that were aggregated at the damage. In gamma irradiated cells, the damage is broadly distributed throughout the nucleus, but when the cells were irradiated with heavy ions, you see streaks or paths of these lesions across the cells. So these foci of gamma H2AX have become very popular to measure as a surrogate for DNA double-strand breaks. They're very sensitive to radiation damage. You can see them after doses as low as 200 milligray, 0.2 gray. And you can also measure repair, because if you induce these very quickly after, or see them very quickly after radiation, if you wait a while, those foci start to disappear. And that's representative of the repair that's going on in the cells that removes the gamma H2AX foci. And those foci are produced in a linear dose response curve initially that is consistent with the same slope as we got from pulsed field gel electrophoresis. So this is an assay that's a good surrogate for measuring DNA double-strand breaks. But we always have to be a little careful, because it really is not directly measuring double-strand breaks. It's measuring the response of the cells to double-strand breaks by producing these phosphorylated histones. And so other things can produce them. So you always kind of got to be careful when you read the literature about what the experiment was. You also need to be aware that this has become a very powerful way to look at other DNA repair processes also. And so now we have developed antibodies that are specific for other DNA repair related proteins. And you can do a similar approach to look at other proteins, and popular ones, and we'll come back in another lecture to talk a little bit about these, are P53, 53BP1, and RAD51, RAD52, etc. And there's a lot of interest in whether these can be used as an approach to study radiation sensitivity of various tumors and normal tissues. So what about numbers? So when we irradiate a cell, I already said there are lots more single-strand breaks than double-strand breaks, and we see that here in this tabulation. For every gray in a cell, there's 1,000 single-strand breaks, but only about 25 to 40 double-strand breaks. Lots of base damages, but as we'll talk about again when we talk about DNA repair processes, many of these breaks, these base damages, are repaired very, very, very quickly and very accurately. Many of the sugar damages produce single-strand breaks, which are also repaired quite rapidly in cells. And so we're left largely, we don't know so much about cross-links, but we're mainly left with these double-strand breaks and single-strand breaks to think about. And something called clustered lesions, which in recent years, we've also become very interested in thinking about. So where did this idea of clustered lesions come from? Well, remember that ionizing radiation is really good at killing cells. Per unit damage that is produced by any kind of damaging agent in DNA, like UV or hydrogen peroxide, much less damage is in the DNA is needed per dose of ionizing radiation. And that has made many of the early chemists think about how that damage must be, there must be something different about the ionizing radiation induced damage. And what was realized was to come back to this energy deposition pattern that I mentioned early on. And so when you have these regions like spurs that actually are about the same size, the same diameter as the DNA molecule, or a little bit bigger regions of ionization clumps called blobs that are about the same size as a nucleosome. So think what would happen if these spurs and blobs landed with lots of energy, lots of ionizations right on the DNA. And in fact, that's how we came to think about these so-called clustered or complex lesions, where the idea was if we look at tracks of ionizations in a Monte Carlo simulation going through water, you can see each dot is an ionization, and most of them are kind of widely spaced. But at the ends of the tracks, and sometimes in the middle of the tracks, there's these clumps. And if these clumps of ionizations land on the DNA, now instead of producing just an isolated base damage or an isolated single strand break, you have double strand breaks or double strand breaks with another nearby damage, base damage, or single strand break. And those clusters of damage are going to be much more difficult for the cell to repair accurately, and therefore they're going to be more lethal. And so the idea is that we know from Monte Carlo calculations that clustered lesions are regularly produced in DNA. They can be very complex. They can include base damages and single strand breaks as well as double strand breaks. And they are very important to increase with LET. And again, we'll come back to that later. So the theory is that these clustered lesions are really much more difficult for a cell to repair accurately than single lesions. And so they can block DNA replication. They can cause a loss of genetic integrity. They could be misrepaired, resulting in more lethal damages like double strand breaks. And so we think biologically that clustered lesions are really the most important. But the problem we have is that clustered lesions are really difficult to figure out how to measure accurately in the lab. And so in theory, we think clustered lesions are really important, but we don't really have good measures of how many of them are produced. And so in the end, we mostly talk about DNA double strand breaks, which is the simplest clustered lesion. And we'll think about those in damaging cells. We've talked a lot about this DNA as if it was an isolated molecule in cells, but of course, you know, it's wrapped around histones and it's organized into chromatin fibers. And when the DNA, when the cells go into mitosis, that DNA gets condensed. And all of these structures and all of the different proteins that are attached around the DNA are also very important for changing the radiation sensitivity of the DNA. Whether or not the DNA is openly transcribing or openly replicating can change its radiation sensitivity. So we have to really also keep in mind that chromatin structure is important. And I'm just going to give you one example because of its clinical relevance, and that is thinking about this chromatin condensation and the presence of histones. Excuse me, just a minute. Maybe some of you heard about using in the clinic histone deacetylase inhibitors that are radiation sensitizers. Well, one of the things these molecules are doing is changing the histone structure, chromatin structure in cells. They can change how condensed or how open the chromatin is. And at least in theory, if you open up the chromatin, there should be more free radical reactions, and it should be more radiation sensitive. And in fact, that's what we think the histone deacetylase inhibitors, the HDAC inhibitors, do. And they're very good radiation sensitizers, shown here by looking at cell survival curves, where you get much more cell killing in the presence of the HDAC inhibitor, and in a tumor, where you can get radiation sensitivity, much slower tumor growth in the presence of radiation with the drug. So an important application in the clinic of chromatin structure and changing the radiation chemistry by using a drug that we know can be used in the clinic. So I've said a number of times, DNA double strand breaks are most important for cell killing or clustered lesions. And this is the kind of data that show that, where we're looking at cell survival, cell killing as a function of increasing DNA damage under many different radiation conditions, and you get a common relationship. It doesn't tell you cause and effect, but it certainly tells you that there's a very close association between DNA double-strand breaks and cell killing. And so, although we know double-strand breaks are really very important, and we often say they're the most important, theoretically, we think clustered lesions are probably the most important damage for cell killing. But remember, that's much of that is based on theory, not actually being able to measure those clustered lesions. So we've been talking about DNA damage. It can be not repaired. And next time, so another time, we'll talk about repair processes. But if that DNA damage is not repaired, then all kinds of bad things happen in cells. And I'm just going to finish up by talking a little bit about chromosome aberrations that result from that lack of DNA repair processes in cells, or misrepair. So chromosome aberrations. Remember that chromosome aberrations are those damages that we see in mitotic chromosomes when the cells have gone into mitosis and condensed to form these characteristic mitotic chromosomes, and we spot things that don't look right, the chromosome aberrations. But those chromosome aberrations, remember, are not only telling you about the initial DNA damage that occurred when radiation interacted with the cells, but also whether it was misrepaired or not repaired, so that it resulted in a chromosome aberration. And geneticists like to talk about two general types of chromosome aberrations, the so-called chromosome aberrations and chromatid aberrations. And generally, what is important is the time at which ionizing radiation interacted with cells in terms of the cell cycle distribution. And so basically, if cells are irradiated before they have replicated their DNA in the S phase, so they're irradiated in G1, then you get chromosome aberrations because the damage occurs before DNA replication, and therefore the damage gets replicated. And so the damages you see occur in both chromatids of the chromosome when it goes into the condensation. So you see things like translocations from one chromosome to another. You see deletions where bits of the chromosomes have come out of the chromosome. And so these are reflected in both chromatids. If the DNA has already undergone synthesis, so at late SG2 phase at the time of irradiation, then you can end up with damages that show up in only one chromatid of the condensed chromosome. You can get really weird looking structures that can be produced by interchanges between the chromatids in that given irradiated chromosome or between two chromosomes. And so you get interchanges, and you can also get simpler structures like deletions. Let's look at some examples. Here's some examples of chromosome irradiations. Very commonly produced by ionizing radiation are dicentrics, where two irradiated chromosomes came together to misrejoin and therefore now produce this complex structure. Or a single irradiated chromosome can fold back on itself in a ring structure, or tricentrics where three chromosomes came together. And not uncommonly, you see fragments, little pieces of chromatin that were produced from deletions within or from a single chromosome. And here's some examples of how you can produce that deletion, where two chromosome breaks resulted in a damaged chromosome, but also with these little pieces of chromatin that can go out into the cytoplasm and be isolated DNAs, as we'll see in a minute. Or you can get two breaks that result in the chromosome bending back on itself and forming a ring structure. These dicentrics where two chromosomes come together to form a single dicentric chromosome, are quite characteristic and quite easy to see in irradiated cells. And so for biodosimetry, for example, for understanding whether an individual has been exposed to ionizing radiation, really a good approach is to look for dicentrics in the circulating lymphocytes of the individual that may or may not have been irradiated. So dicentrics are really a great measure of DNA damage. Some other chromatid aberrations now are these really complex structures where damage in two chromatids or in a couple of chromosomes come together to form these weird complex exchanges and quadraradials. So ionizing radiation produces some very characteristic structures in chromosomes, dicentrics, rings, fragments, translocations. Some of these can be symmetric, which means there's no loss of genetic information, and those might be stable and be seen even years later in irradiated cells. But many are asymmetric and unstable because genetic material has been lost from those cells. And typically, those cells are going to be lethal. These fragments that I've mentioned over and over again have also become a good way to look for irradiation in cells, because when the cells separate at mitosis, if these fragments don't have a centromere in them, they produce what's called micronuclei out in the cytoplasm. So they're little bits of acentromeric chromatin that can be seen in the cytoplasm. This is an example of nuclei after irradiation that have been stained. And I hope you can see there's a micronucleus here, there's one there, there's another one out here. So these micronuclei are a relatively easy way to measure a surrogate for chromosome aberrations. And in the last few years, we've come to recognize that there really might be an important biological underpinning for these micronuclei, because it's been shown that in some cases, they're important for stimulating immune responses in irradiated cells. So they can initiate the C-gas sting-dependent pro-inflammatory immune response in cells. So lots of important things about micronuclei. Nowadays, mostly we look at chromosome aberrations by staining them in a process called chromosome painting, fluorescence in situ hybridization, excuse me, where we can stain chromosomes with chromosome-specific fluorescent probes or even specific regions of specific chromosomes, and really be able to much more easily see things like translocations, deletions, and all kinds of chromosome aberrations that show up with chromosome painting. If we look at lots of damages in cells and try to count them as a function of radiation dose, what we see is that the dose response curve for radiation-induced aberrations is what we call a linear quadratic dose response curve, which means at low radiation doses, it's linear. At higher radiation doses, it becomes quadratic or dependent on dose squared. And this shape for the dose response curve becomes important when we think about radiation-induced cell killing and survival curves that we'll talk about in a later lecture. So keep this linear quadratic dose response curve in mind. And we'll talk later about dose rate effects, how increasing or decreasing radiation-induced dose rate allows for repair during the radiation exposure, and that means less damage. And we'll see that in a subsequent lecture in irradiated cells, looking at cell survival, but we also see that in chromosome aberrations, where at very low dose rate, far fewer aberrations are produced than at a higher dose rate because of the repair that occurs. And another effect we'll talk about later that we can also see in chromosome aberrations is that of high LET, where the linear quadratic curve that we saw at gamma rays and X-rays becomes much steeper and straight when we look at high LET radiation because of lack of repair. So you don't get the repair of those radiation-induced DNA damages. That's looking at dissects. And just like there was a really good correlation between double-strand breaks and cell killing, we see the same thing with chromosome aberrations. So you get a loss of colony formation, a loss of clonogenicity that correlates very well with the production of chromosome aberrations. So just to conclude, a few take-home messages that things I've tried to emphasize today. At low LET radiation, we get lots of indirect effect because of the production of that damaging hydroxyl radical in cells. And that hydroxyl radical reaction produces lots of DNA damages that we can talk about. And the most common assay that you will see in the literature is that nowadays for looking at the induction of foci of DNA repair-related proteins. Chromatin structure is important because we can modify that to modify the radiation damage. And there are very characteristic types of chromosome aberrations produced by ionizing radiation. And we'll come back next time to talk about that linear quadratic dose response curve that is really important when it's produced by ionizing radiation. And with that, I see there are a few questions. How do you want to do this, Bobby? Can you bring the questions up? Should I stop sharing my slides, or what do you want to do?
Yeah, I think we can read the questions. So I can read for you if you want, and then you can.
That way we can
answer.
go back to the slides if we need to help out with answers.
Yes, sure. You can back to the slide if you need to answer with the slides. So we have now we are open for questions. So we so far we have two questions. First, I will read first from Kashmal Mafazi Shahril. I think he's a resident from Indonesia. Thank you for the opportunity, Professor Held. I have a question as follows. Do all of these effects of ionizing radiation occur at the same proportion between each other, or any of them occurs and produces a more dominant effects than the others?
Oh no! Remember that table I showed you that gave quantities for the production of these various types of damages? So there are many more single strand breaks and base damages produced than double strand breaks, but cells can very easily and accurately have great repair capacity for handling the base damages and the single strand breaks. And so those get repaired and aren't really important ultimately in what happens in cells in most cases. And so the important ones are these double strand breaks or the clustered lesions that, even though they're not produced in very high yields compared to the other things, they're not very accurately and readily repaired. So in fact, there are really big differences. If you want to look at the chromosome aberrations, for example, we know that any of these chromosome aberrations like rings and dicentrics that are really difficult for a cell to divide accurately. You can imagine at mitosis, a cell that's got a ring isn't going to be able to go accurately. The two pieces of it are going to have to break for the cell to go to the mitotic spindles. The cell goes through mitosis. So again, these are going to be really lethal aberrations. But on the other hand, some of the things like translocations, if the amount of damage is if no genetic damage is lost because it's a symmetrical translocation, then in that case, it might not be a lethal aberration. So there certainly are very different levels of severity of these different lesions, either at the DNA level or at the chromosome aberration level. Does that make sense?
Yes, think you already answered very complete. So we can go to the next questions. This is from Teguh Muhammad Khalid. Good afternoon from Indonesia. Thank you for your explanation. My name is Khalid. Currently, I'm a resident from Indonesia. I want to ask a few things. First, regarding phase of cell cycle and its impact on radiation effect. We know that high dose accelerated fraction can also induce cell damage and also promote cell immune complex. How is it different from conventional fractionation? So I need to read all of you want to answer one by one, Dr. Kathy.
Well, let's answer them one by one.
Okay.
I mean, clearly, cell cycle changes the response. Cells are much more sensitive to radiation in certain phases of the cell cycle, like G2M, and also repair changes through the cell cycle. But I'm going to really get into some of that in a later lecture. So would it be okay if we defer that question until if I see if I explain it clearly in a later lecture? Because otherwise it's going to take a long time to explain it right now.
Yeah. So Dr. Khalid, we will have those lecture in the next in lecture series of Dr. Hell. So I think also the question number 2, how is the radiobiology different in very high single dose compare SR compared to cell damage from conventional? I think it is also in your other topic, right?
Yeah.
Yes.
We'll talk about that. Yeah, we'll talk about that later. Yep.
Yes. So all the attendees, you have to follow this lecture so you don't miss only one lecture.
Yeah, everybody's going to have to come to every lecture. Right.
Yes. And number 3, regarding tumor doubling time that is differing from cancer types, will it affect our fractionation schedule?
That's not again, that's a really information we'll touch on in later lectures. It's not a simple answer because it depends on many factors, not just doubling time that's critical. So again, I think I'd like to defer that till after a later lecture and see if we can get to it then.
Okay. So you might find the answer in the later lecture.
I think so.
Next, we have a question from Dini Alyani. Thank you for the opportunity. How cisplatin can work effectively in using the double strand break? Does it has specific target structure in chromosome? Just what you have just mentioned with agent targeting histone specific.
I'm sorry, I didn't catch the whole question.
So about giving the additional of sensitizer like cisplatin, how does it can effectively induce the double strand break? I mean, if it is the additional of chemo.
Cisplatin, it depends on the radiation. Remember that chemotherapeutic agents act by many different processes. They're not all the same. And so it depends on what kind of DNA damage they induce, whether or not it can interact with ionizing radiation. And specifically to think about cisplatin produces crosslinks. So it increases the DNA crosslinks. And so you can get in theory, you get an interaction between radiation and the cisplatin induced crosslinks that can occur. And in fact, let me go back some more. The kinds of forget where the figure is. It can produce that one. It can produce crosslinks that are both intrastrand and interstrand. And so it's a different requires there's an overlap of the repair processes that are needed for both repairing DNA double strand DNA induced damages and the crosslinks that are induced by cisplatin. Other kinds of chemotherapeutic agents cause other kinds of DNA damage. And so we can't really make any generalities. You got to think about every chemotherapeutic agent and its potential interaction with radiation on a molecular basis individually. Did that hopefully answer that?
Yes, Dini, I think it's answer your question, Dini. The keyword is cross-link. Okay, next we still have more 2 questions. This next question from Joshua Ho. So Joshua Ho, thank you so much for the lecture, Professor Catherine. I'm asking as a physicist. So first question from Joshua, would irradiating bone have different percent of indirect or direct damage to DNA as opposed to 70 or to 30 in water?
I'm sorry, irradiating
Bone.
Well, even bone has, cellular structure. So there's predominantly water, even in bone structure. So I don't think it's an interesting question. I don't think there's significant differences in terms of the basic radiation chemistry that would go on. There might be a little more direct effect, but I think there's still predominantly water in those tissues.
Okay. So the second question, does DNA react differently to different free radical in terms of DNA damage?
Yes, the chemical reactions that go on are quite different. I showed that just there. In terms of the OH radical, reacts differently with different parts of the DNA molecule. And remember, the OH radical is an oxidizing species. The aqueous electron and the hydrogen atom are reducing species. So the reactions that they do are going to be reduction reactions instead of this kind of an oxidation reaction. So they are quite different reactions. But because of the environment, the redox environment in cells, they very rapidly get essentially back react, they get negated. So biologically, they don't produce the OH radical and the aqueous electron biologically don't produce damages that go on to form free radicals in DNA that then go on to degrade and produce the DNA damages.
Okay.
If we look at that scavenging experiment here that I showed you, this with the OH scavengers, if we did the same experiment with a chemical that reacted, for example, very well with the aqueous electron, you would see very little protection. It wouldn't make much difference, unlike what we see with the hydroxyl radical. So they're not protected by removing those other free radicals.
Okay. So we can go to the next question from Joshua. Can foci tests like 53BP1 be treatment efficacy proxy for patient undergoing radiotherapy?
Well, that's a theory. There are people who would like to do that, would like to be able to look, for example, at the radiation response, production, and or the repair of these foci in samples from tumors before you start to treat or partway through the treatment, that you could get an idea of how well the tumor is responding to the radiation treatment. At this point in time, that's really a theory and really being looked at in some clinical studies where people are trying to understand whether they can use that as a predictive assay for how they should treat tumors, or whether a tumor might be particularly repair defective. And that might mean you could not have to give as high treatment if they're intrinsically more radiation sensitive. So it's a great idea. It's not yet shown that it's going to work in the clinic, but it's certainly been looked at.
Okay. So I think it answer your question, Joshua. So we can proceed to the next question from Frizky Ramadan. Thank you for the opportunity. My name is Frizky from Indonesia. I would like to ask question, are there advancements in radiobiology research that could potentially lead to more targeted and less toxic radiation therapies for patients?
Oh, lots of them. I mean, we'll talk about a lot more things coming up, but you should know that from your clinic. There's a lot of clinical trials that are attempting to look at new combinations of drugs, looking at all the exciting changes that are coming now with immunotherapy, with changing to ultra high dose fractionation, for example, flash radiation therapy, with using protons, with using other high LET radiations like carbon ions. So there's many, many exciting new approaches that are being looked at in all kinds of clinical trials all over the world.
Yeah. Okay, thank you. So we go to the next question from Vindang Huang. Thank you, Professor, for your lecture. I have a question. What is the difference in DNA damage between normal cells and cancer cells? Thank you very much.
Well, there's no, in the initial amount of damage that is induced by ionizing radiation, there's no difference. These kinds of damages that I've been talking about today, from the DNA damages, the chromosome aberrations, are seen in both normal cells and tumor cells. Now, we certainly now recognize that some tumor cells are defective in specific DNA repair processes, and that might make them more radiation sensitive, especially if we could identify those and figure out how to exploit those differences. Normal cells, by and large, unless it's an individual, very, very rare individual, for example, that has a radiation sensitivity syndrome like ataxia telangiectasia, generally normal cells are very repair competent. So what we are interested in trying to understand is the repair deficiencies in various tumor cells, and how can we identify those and exploit them for better radiation therapy. Lots of research there to be done. Opportunities for you folks.
Yeah. Okay. So we go to the next question. This is from Melida. Given the challenges in measuring clustered DNA damage, as what you point before, are there any indirect or surrogate markers that researchers can use to infer the presence and extent of clustered damage? How reliable are these proxies in clinical practice?
Well, not at all. Nothing that we can use in clinical practice. There are approaches that people have tried to look at them. And in particular, for example, since we're on the slide, the gamma H2AX foci is a potentially a way of looking at the clustered lesions, because a lot of studies have shown that, for example, with high LET radiations, the clusters, the foci are bigger. So it suggests that the clusters are bigger. And we know that with high LET radiation, that there's much less repairability. We can theorize that there's much bigger clusters. That was shown here. You can see, it's a little hard to see in these figures, but the generally under same conditions, if you'd irradiate cells with high LET radiation, these foci are going to be bigger than they would be when they're irradiated with gammas or X-rays. So that's taken as support that there's differences. There's really not other good ways that I'm aware of to measure clustered lesions directly. And that's an area that it would be great to have some advances in our research.
So I would like to ask about these slides comparing gamma rays and heavy and the higher LET. So how about in proton? The proton LETs slightly just higher than gamma rays. Does it also create the same like heavy ion, or almost same like gamma rays?
Well, first off, keep in mind that the protons, as you've already alluded to, the protons that we use clinically are not high LET, they're low LET. So much more like in their biological effects, like gamma rays and X-rays, than they are like iron ions or carbon ions. And in fact, if experiments are done under identical conditions, by and large, the foci from protons are more like gamma rays than they are like iron. But there is increasingly, again, some research that is showing that some protons of irradiation in some types of cells, particularly maybe as a way to identify repair defective tumor cells, are going to be more sensitive to protons than you would expect with than you see with X-rays and gamma rays. So you might see slightly larger foci. And there have been some attempts to try to look at that experimentally. Some of my own colleagues at Mass General have been involved in some studies like that to try to look at size of foci. But at this point, it's not definitive. It's certainly suggestive that the foci are a little tiny bit bigger, but it's not dramatic differences. And it will also depend on the repair capacity of the tumor cells you're looking at.
Okay, thank you. So we have another two questions here. One is from Ferdinand Maubere. Thank you for the excellent lecture, Prof. My name is Ferdinand, resident also from Indonesia. Back then, I thought radiotherapy worked like a bomb, affecting both normal cells and cancer cells somehow equally, hence the side effects accompanying the RT treatment. But through radiobiology lecture like this one, we learned that radiation works like a sniper rifle, shooting right into the lethal killing point of the enemy, which is the DNA. The question is, is DNA damage is the only beneficial killing effect of irradiation to cancer cells, because it is indeed the most effective and efficient one, or it's only the tip of the iceberg, or what irradiation can actually do for cell killing? I'm interested in your insight on this, and there is a future opportunity we can have more diverse way of killing cell through irradiation.
That's a good question. Certainly, most of the evidence that we have suggests that most cell killing occurs because of DNA damage. If you just look at those good correlations that, where'd it go? Those correlations I showed between DNA damage and cell killing. Now I can't find it. Where'd it go? DNA damage and cell killing and chromosome aberrations and cell killing. I'm going the wrong way. I can't talk and look at slides at the same time. When you look at those correlations, where's the slide going on? That one. These kinds of correlations, it makes it clear that much of the cell killing is because of DNA damage. And that's probably what's most important in tumor cells. Remember, in tumor cells, in radiation therapy, you want to prevent that cell from growing anymore, from forming a colony, from continuing to proliferate. And that really is where DNA damage is important. In normal tissues, it's probably, and also in some tumors, it becomes more complicated. And to kill a cell, DNA damage is clearly important. But in normal tissues, remember, you got lots of cells that aren't dividing. And so we must be seeing damages that are not simply because of cell killing. And that's where we start to recognize that a lot of the late responding normal tissue damages and even acutely responding normal tissue damages are not simply a result of cell killing, but are very complex interactions that include stimulation of inflammatory responses, of immune responses that are effects on the whole tissue, not just killing individual cells. So it becomes a lot more complex. Now we recognize that happens in tumors also. But the immune stimulation happens, the inflammatory reactions can occur to help get rid of tumors also. But in the end, to kill all of the tumor cells, I think we still think that you got to kill those cells, and that requires DNA damage. In normal tissues, you get a lot of damage that is because of the stimulation of inflammatory reactions, of immune reactions, and it's not just as simple as cell killing. And again, I think I'll touch on that in a later lecture. But that's a really important question.
Okay, thank you. So, Dr. Ferdinand, you have your answer. Next, I think this is the last question, or is there any more question? Oh, okay. From Sean Yi Lim, thank you for the lecture. Since alcohol acts as a radical scavenger, will it cause radiotherapy to be less effective if a patient drinks alcohol during treatment?
No, because you can't drink enough. It'd be toxic if you drank that much. Those were cells in culture, and you'd had to put in lots of alcohol. So no, it wouldn't happen. What we remember that already in cells, there's all kinds of other things that can act as scavengers within the cells too. There's all the proteins, there's all the sugars and amino acids and so forth also in cells. So we've already got normally a lot of protection by radical scavenging. So these are really designed experiments. You wouldn't be able to drink enough alcohol to get that much radiation protection. So you don't have to worry about that from a radiation point of view in your patients.
Okay. So by the time the patient is having radiation, the doctor already told the patient to stop drinking alcohol.
Well, I would hope so, but it won't matter.
Yeah. So the next question from Bin Hussein Ainuddin Husairi. Hi, good evening, Prof. Thank you for the lecture. Ainuddin from Malaysia. My question is, does antioxidant has any significant effect to interfere with cell killing in tumor cells? Does antioxidant has significant interaction with the free radical produced that can cause radiotherapy less effective?
That's another really good question, and I don't think we clearly know the answer. We know that antioxidant processes are certainly important in modification of cellular responses. Exactly how that works in patients when we're irradiating their tumors and their normal tissues, we don't know. There are, again, clinical studies going on where people are looking at taking antioxidants and how that changes the metabolism in the tumors and in the normal tissues. And there's some suggestions of antioxidants being protective. There have been suggestions, take lots of vitamin E and vitamin C, and that might help protect the normal tissues. You don't want it to protect the tumors, obviously. So really important questions that we don't know the answer to, but in theory, yes, this radiation chemistry is all oxidative reactions. And if you can prevent those oxidative reactions, it would matter. And you might know, some of you from some of the literature, there's interest in looking at superoxide dismutase mimetics. I mentioned superoxide dismutase early on, remember? The superoxide radicals produced in irradiated cells and tissues. And we have some clinical studies going on to look at whether superoxide dismutases can be used to protect normal tissues. And in fact, there's some good clinical data coming out that suggests, in fact, that antioxidant process can protect under some circumstances with superoxide dismutase mimetics. So lots of exciting things to look at in those looking at antioxidants.
Right. Okay, I think that's all of the questions here. Oh, okay, this is the last question because of the time. From Hatran, does radiosensitizer make normal surrounding tissue be killed more than usual?
It depends on the radiation sensitizer. There are certainly some drugs that we know can interact with radiation to increase damage to normal tissues. No question about it. Again, the chemistry, if you're looking at sensitizers that work by modifying just the chemistry, radiation chemistry, that's going to be the same in tissues in normal and in tumors and in normal tissues. So the trick is to be able to somehow keep the drug in one or the other, or keep the radiation in the tumor and not in the normal tissue by focusing it better. It's always trying to exploit the differences between the tumor and the normal tissues. A simple sensitizer that works in any kind of cell is going to work on both, is going to sensitize both normal tissues and tumors. We have to find something that's different between the normal tissues and the tumors that we can focus in on when developing radiation sensitizers. And we'll talk in another lecture very briefly about hypoxic cell radiation sensitizers. There was a lot of interest in developing them years ago because of the belief that there were hypoxic cells in many tumors that were not present in normal cells. And so that would be a way to exploit that difference if we could get those drugs selectively into the tumors, into the hypoxic regions. So it all comes down to exploiting differences to be able to develop sensitizers or protectors the other way around. If you could get a protector, like we were just talking about, the superoxide dismutase mimetics, that's just in the normal tissues, that would be good too.
Yeah, right. Right. So I think that's all for the questions and answer. I think there are 11 questions for tonight, and very active from the attendees.
Well, that's great. And you have for my slides my email. If anybody has more, oops, I went too far. Well, anyways, you had in my last slide. You can look at my emails, and be sure, don't hesitate to email me with more questions.
Right. Also very kind, Dr. Held. So ladies and gentlemen, guys, I think this is the first lecture of a series of 6 lectures. So wait for the second, third, fourth lecture. There are many more to come from Dr. Held. Dr. Ben, Dr. Enrico, do you have any comments?
Go ahead, Ben.
Ben,
Your mic.
Yeah.
I talked earlier, I think it's Dr. Enrico's turn. I was just impressed. Those are questions that the residents might be afraid to ask us, or they consider asking us as not really worthwhile, because those are questions we might not be able to answer satisfactorily. So thank you for that. Dr. Enrico, do you have anything to add?
No. Well, of course, the lecture was quite excellent. Congratulations, Dr. Held. We
Thank
would
you.
really appreciate it. You know, the ASEAN countries are not really that rich, so to speak. And to have an expert like you give a lecture to us, we're over about 200 plus participants, is really of a great help to the society. And we really appreciate your help in doing this. Thank you very much.
Well, it's my pleasure. Thank you for inviting me. I enjoy very much teaching and like interacting with young people. So I'm pleased to be here.
Okay. So before we close, Dr. Dika, can you share the schedule for the next lectures from the poster? So as I mentioned before, we will have 6 series of lectures. This is the first, and the second will be 1 month from now. We are trying to be in the the 4th weeks of each month. You can see from the monitor, you can see the second will be at 28 November, and the third is in December, and so on, so on. And please remind that this lecture is every in Tuesday. But for the time, I think for the next lecture, the second lecture, we will announce later, because there are some change in zone, in time zone or daylight time. So it will be earlier 1 hour compared to now, or later. So we will inform to you all later. But the link only one time registration. So in the second or third or fourth lectures, if you have already registered, and then the link of the Zoom will automatically be sent to your email. So thank you, Dr. Dika. You can stop share. So I think that's all for tonight. Dr. Hel, do you have any comments?
No, thank you very much for inviting me, and I look forward to seeing you again in a month.
Yes, thank you. See you in month on Tuesday. Thank you, everyone. Thank you for
Thank you.
attendees, everyone. Thank you, Dr.
Thank
Ben
you.
and Dr. Enrico. Everybody, bye-bye. Good
Thank you so much,
morning.
Dr. Hel.
Take care. Bye-bye,
Evening.
everyone.
Bye-bye. Have a nice day.